Hi, today I want to talk you through the Amylase lab investigation. So the whole point of this lab is to see what happens with the saliva in our mouth. Saliva is mostly made of water, but it also has enzymes in it. One of those enzymes being amylase. And the enzyme's job is to help start that digestion in our mouth. We know physical digestion starts with our teeth, but we're trying to see if chemical digestion happens in our mouth where something new is formed. So in this lab, we're seeing what happens to the complex carbohydrates in the foods that we eat in our mouth when it encounters saliva. So what I started out with is just some rice in water. So the rice is a very starchy food, has a lot of complex carbohydrates, no glucose, so it's just starch. We want to see what happens when this rice is exposed to amylase, which is an enzyme found in our spit. So what I did is I took another batch of rice and I mixed it with water, but I added amylase to this one. So this has amylase in it to simulate what would happen in your mouth with the spit when it comes in contact with the rice. We're doing an investigation to see how the food by itself is different in terms of the molecules it has in it compared to the food that has been exposed to the amylase. So what we did in the lab, we had our 4 microcentrifuge tubes. We labeled them A, B, C, and D. A and B received liquid in it that was just the food. It was just the rice and water. C and D had liquid in it that was the food plus amylase. We wanted to see how the food reacted to the amylase. Just like in our first lab, the dialysis tubing lab, we used 2 indicators. We used iodine to look for starch. It's brown on its own, turns black when it is exposed to starch. And Benedict solution, it's blue on its own and it turns orange when exposed to glucose, but only at high temperatures. So what we did is we took our samples. Remember, A and B was liquid that just had the food in it. We took A and we tested it with iodine to see what would happen if there was starch. And we found it turned black. The iodine detected the presence of starch in the food molecules without any amylase. So food on its own turned black means that there's starch in it. Then we tested tube B, which was food by itself, with Benedict solution to look for glucose. Put it in the hot water, and we got this, blue, no glucose. So the food on its own has no glucose in it. Then we did the same test with the liquid that came from the food that had been sitting with the amylase in the water, kind of like our saliva. We tested tube C to see what would happen with iodine to look for starch, and we found that it turned a blackish color. Tells us that there's still some starch there. We tested tube D with Benedict solution to see if there's glucose after it's exposed to amylase, and we found it turned orange. That tells us that there is glucose in the rice now because it's been exposed to the amylase. We have just witnessed a chemical change occur. The food on its own only had starch in it. However, the food, when it's exposed to amylase, allowed for a chemical change to occur where some of the starch, the complex carbs, broke down into glucose, a simple carb, which is a new food molecule. So this is an example of a chemical change that is happening in our mouth as a result of the saliva.